This presentation will provide a brief overview of an emerging bioengineering device called the organ on a chip. We will cover what organ chips are, why they are important, and a few examples of how chips work and how they are used.
Organ chips are small devices about the size of a USB drive, made with clear polymer and microfluidic channels that model human fluid passageways. The chips are created with computer chip technology and contain real human cells within the passageways. The channels in the chip allow for the generation of mechanical strain on the cells. This creates an excellent model for the real strain and environmental conditions cells experience in a real organ.
Therefore, an organ chip is a tiny but accurate replication of the biochemistry, function, and physiology of human organs.
Currently, drug research and development is highly inefficient and very expensive. The Wyss Institute estimates that it takes $3 billion and more than 10 years to successfully bring a drug to market, and the problem is only getting worse. The scientific community is spending more time and more money to produce fewer and fewer treatments. This issue exists because our current methods of testing drugs for human safety do not work. Our current drug testing tools are human cells in petri dishes and animals. Animals are not a good model because they can be too dissimilar from humans and respond to drugs in different ways. Cells in dishes are not good either because they lack a realistic environment compared to the human body. The cells need to be under mechanical strain in order to truly model cells in a human. Therefore, human organs are great for accurately modeling humans because they can replicate the environment of cells in the body, including the mechanical strain experienced by the cells. This offers great potential for drug testing that is more accurate, efficient, timely, and cost-effective.
One example of an organ chip is the lung on a chip. The lung on a chip has three major fluid channel types. The first two are the blood and air channels, which run parallel to each other and are separated by a porous membrane covered in cells. On the blood side of the membrane are capillary cells, and on the air side are the human lung cells. On either side of these channels are vacuum channels that produce mechanical strain on the membrane and cells. This configuration is a great model of the alveoli in human lungs, which are the tiny air sacs where gas exchange occurs. The lung on a chip is also great for disease modeling. What you're currently seeing in the video is an accurate representation of the human immune response in a lung chip. Bacteria are fed into the airstream where they begin to stick to the lung cells. Then, white blood cells are fed into the bloodstream, and the white blood cells sense the bacteria. They stick to the capillary cells, filter through the porous membrane, and enter the air channel. Once there, the white blood cells attack the bacteria and prevent infection.
The Wyss Institute is an academic research facility at Harvard University that developed organ chips. The Wyss Institute has currently developed the following chips for commercial use: brain, colon, duodenum, kidney, liver, and lung. Under development, the Wyss Institute is also creating a gut chip, heart chip, bone marrow chip, and skin chip. Organ chips can be used in a variety of capacities, including pharmaceutical research, disease modeling and study, and the testing of common chemicals. They can also be used in applications including bioterrorism, radiation exposure, cosmetic testing, clinical trials, and personalized medicine.
These incredible tiny chips are used in labs across the country, including large pharmaceutical labs and the FDA. They are used to study a wide variety of diseases and health concerns, including cystic fibrosis, COVID-19, the flu, malnutrition, radiation, space travel, and many others, allowing researchers to provide critical discoveries to global healthcare professionals.
The future of organ chips has many exciting applications to medical research. One of these goals includes creating a human on a chip, which is a collection of 10 plus connected organ chips representing the larger organism. Another goal is to create patient-specific chips using stem cells. These organ chips would be used to create personalized care plans for an individual based on research conducted using their own personal chip. These applications could revolutionize healthcare research and improve patient care worldwide. Until then, thanks for watching.